So everyone, I'm excited to be here. Akeem Gardner, those lights are bright. First of all, May is ALS Awareness Month, so I'm very excited to be able to be here to present on what we're doing in regards to ALS research. I want to challenge all of you. If you know the Ice Bucket Challenge, go home, do the Ice Bucket Challenge, challenge your peers to do that. And we are continuing along with the ALS Association to raise money for ALS research. It's very well needed. Now today I'm going to try something a little bit different, a little bit new. So I'm going to start by asking a question. Have any of you guys watched the movie Oppenheimer? Oppenheimer? Learned the story of the atomic bomb, right? I thought it was a very, very good movie, a powerful and compelling story. One of the things that blew me away, no pun intended, but it blew me away, the striking power of nature when combined with modern science. And just as that film aimed to bring to light a pivotal moment in history, today I aim to draw a similar parallel, but in the realm of medical science to benefit human health. By the end, my aim is to convince you that complex human disease are best solved with the complex but beautifully designed power of nature. Not nature as a destroyer, but nature as a giver of life. And today I'm excited to share with you the story of how I think we get there. Good. Perfect. So to quickly recap, our mission at Caenorhabditis is to develop therapeutics that target inflammation and address unmet needs in neurology. And we're using the synergy of cutting-edge science and proprietary natural ingredients to do just that. Over the last 3 years, we've been exploring uncharted plant molecules and doing a lot of work to validate them preclinically. Our team is young, talented, gritty, and driven. They're such a joy for me to work with each and every day. And we have a science advisory board experienced and proven in botanical pharmaceuticals. Our goal is to target inflammation, the root cause of all disease, because 50% of human mortality is attributed to systematic chronic inflammation. Now, most drugs on the market and the ones that still fail in development still have pretty bad safety concerns. And botanicals, though generally regarded as safe, have efficacy, standardization, and patentability issues. But as the prevalence of disease continues to rise, we at Caenorhabditis see an opportunity to challenge the traditional drug development and treatment paradigm. Now, Cannflavins A and B are two of our star molecules that help us do just that. These two are prenylated flavonoids unique to Cannabis Sativa. They have a flavone backbone consisting of 2 phenyl rings and a heterocyclic ring. They're highly lipophilic, and that allows them to easily accumulate in cells and potentially membrane-bound enzymes and receptors. And they're traditionally found extremely scarce and rare in the plant, in under 0.014% of the plant's biomass. Well, we've developed production technology, patented tech that allows us to be able to produce these as extracts alongside purified molecules that we've been able to use to gain deeper understanding of their mechanisms, how they work against inflammatory and neurodegenerative targets. Today, I'm going to start by laying the groundwork of how these molecules might be beneficial in areas of neurodegenerative disease. Cannflavin A is a dual inhibitor of pro-inflammatory enzymes. It has 30 times the potency of ASA, the active ingredient in aspirin, but it doesn't have COX-1 activity as it works through 5 alone. So a higher safety profile is expected with repeated and chronic use. Now, though this initial discovery was made in vitro in the mid-1980s, no one's produced enough of this molecule to be able to validate the original assays with botanically derived ingredients, as we have here. Or they haven't moved things forward in vivo, not in a human or in a mouse until now. What we were able to do was do an LPS-induced acute respiratory distress model that was able to show that Cannflavin A was able to reduce cytokine storm markers and ameliorate the pulmonary function in a statistically significant way. We have other data not shown on the slide, but it shows that we were able to decrease MPO activity, which is a measure of inflammation, and a reduction in the cytokine known as CXCL1. Notably, while dexamethasone, I think we can see it here, in this one is what I'm talking about, Dexamethasone was failed to improve the respiratory dysfunction, improved by LPS. Our Cannflavin A successfully restored the respiratory function in these mice. So then we continued to look at how these molecules could help, and what we used here was C. elegans in a model due to their genetic and behavioral suitability for simulating human neurological conditions. In these studies, compounds were tested at a concentration of 20 micromolar, with endeavorone serving as the positive control. Over 10 experiments, 3 that I'm going to show you today, our Cannflavins exhibited significant activity, especially in models related to ALS and frontotemporal dementia. This suggested to us a valuable direction for future research into their potential benefits. And this is what brought us to ALS, otherwise known as Lou Gehrig's disease. For any of you not familiar, ALS is a rare, debilitating motor neuron disease with rapid progression and average mortality coming just 2 years after diagnosis. There's currently no cure or treatments to manage symptoms, as the disease is very complex with a multifactorial nature of neuron degeneration. There's a need for new treatments to meet the unmet needs of patients, as every 90 minutes, someone in the US is newly diagnosed with ALS, and every 90 minutes, someone with ALS passes away from the disease. It's hard to understate just how much of a challenge this disease is and how much of a challenge it's been to modern science, as it's completely heterogeneous, meaning it manifests differently in each patient, and it's governed by a unique combination of genetic, environmental, and biological factors. And there's so many different things going wrong in the motor neurons once they start to degenerate. The disease also exists on a spectrum with other diseases like FTD and Parkinson's, so symptoms are often misdiagnosed, leading to a challenge when it comes to treatment. Traditional drug therapies are often designed to target a singular pathway, and these continue to fall short in effectively treating the multifaceted disease. Now, one would think with our new technology and our new ways of making medicines today, over a 150-year history of this disease, we would have some traction towards a solution. But nope, life expectancy back then was 2 years post-diagnosis, and it's still the same today. And the options that we have on the market today are no good, as patients are left with the options of taking multiple drugs, multiple pills, various solutions to help them manage a debilitating quality of life. Even in today's news, we still continue to see failure after failure after failure in clinical trials. Most recently with Amylyx voluntarily withdrawing their medicine due to a failure after phase 3 clinical trials, even though both the analysts and the company thought that this could be a blockbuster drug. But once again, patients were let down and disappointed after promising phase 2 data because it didn't work. And this is just top of the news over the last 12 months. A lot of failures from a lot of pharmaceutical companies. Now, this has led to industry experts calling for change in how we approach the ALS diseases. It's not just one disease. You have to treat the illness in multiple different ways. And someone in the room here with us had a similar calling. He had been talking about it for many years. This was Dr. Ethan Russo, our senior medical advisor and CEO of Credo Science, where part of his business is formulating novel botanicals that could be developed pharmaceutically. A few years ago, Ethan began to tell me a story of a patient that he had first treated when he came to practice, diagnosed with this terminal disease. And this patient refused to be treated with the traditional pharmaceuticals. His choice of therapy was cannabis. And as Ethan will tell you, over 10 years of seeing this patient, he never got weaker. He also told me of his personal mission to do something here for this patient population, as he too felt the pain of watching a loved one pass away from the disease. So as Ethan and I started to talk, this is where things started to change for me, as Ethan let me know about a patented formulation he had been working on throughout his career that he knew would be able to serve this patient population. So the first thing that I needed to catch up on was learning the full potential of the cannabis plant, one of nature's greatest medicinal plants. With respect to ALS, it's important to note that there's much anecdotal evidence reported of the plant's utility, as patients suffering from both ALS and other neurodegenerative diseases have used it in various forms since antiquity to manage their symptoms. The second thing I had to learn was all the advantages of the botanical drug. These are multi-target therapies that are made up of well-tolerated ingredients that we as humans co-evolved with in nature. They can be delivered in a uniform product, and as many of you know, there are streamlined advantages in developing botanical drugs, with regulators around the world recognizing the long history of traditional use that we have for these medicines. And lastly, patients, they generally prefer naturally derived medicines anyways versus the synthetically derived ones. They all lean towards the laws of nature. Now, for me, as a law school graduate, I love that I get to lean on the historical evidence of proof of cannabis's utility in human health. Like in 2700 BCE, Emperor Shenyan wrote that protracted taking may make one fat, will make you strong, but you'll never go senile. In the 1200 CE, writings from the Anandakhanda shared that in combination with celibacy, those who used cannabis as part of a rigorous regime could live 300 years free from any disease and signs of old age. Maybe worth it, maybe not, I don't know. And in the 9th century, Al-Kindi famously wrote, "Hashish eases the muscles of the limbs and what flows." Now, more recently, the bioactive molecules from the plant have been pharmaceutically developed and standardized to treat neurological diseases like multiple sclerosis and Dravet syndrome with commercial success. And researchers have long investigated how the key constituents from the plant may be useful for helping patients suffering from ALS. One of these studies was a randomized controlled clinical trials of nabiximols, trade name Sativex. 60 patients in the study, 29 took nabiximols, all of them looking to treat their spasticity. And here, not only was the drug tolerable, but patients showed statistically significant improvement when compared to placebo at 2 of their endpoints, the modified Ashworth scale for spasticity and on the pain numeric rating scale. But the trial wasn't powered strong enough to show statistical significance on all the other secondaries, including the ALS functional rating scale revised. Though some of these findings were statistically significant and some were not, what's key here is that Sativex was not optimized for the ALS patient. This is exactly what we have the ability to do with CNR 401. This is our botanically derived cocktail mixture specifically designed to harness the powers of nature, standardized in a way that will meet the requirements of regulators like the FDA. And just like a nuclear bomb doesn't rely on a singular reaction, but a series of simultaneous reactions that amplify each other to produce a massive synergistic effect, similarly, 401 isn't just us throwing together a bunch of ingredients in a pot or an extract and hoping that one works. It's a calculated strategy where each component plays a specific role, enhancing the overall drug's effect. The beauty of this approach lies in its synergy. Each ingredient in the cocktail is selected not only for its individual effects, but how it interacts with each other. This is crucial because in ALS, where multiple cellular and multiple pathways are impaired, what we have the ability to do is to provide benefit by targeting multiple pathways simultaneously, leading to a therapy that will create a cascade of beneficial effects, mitigating the disease from several angles with each and every dose. Now, I know all this sounds nice, but some may ask, where's the proof? What are the other ingredients? How do we know this approach will work? Well, now I'm going to take you through a little bit about what the last few months have looked like for us and where we intend to go from here. So several toxic agents, such as BPA, BMAE, pesticides, and heavy metals are known to trigger symptoms similar to ALS. BMAE in particular is produced by a symbiotic cyanobacteria and has been linked to ALS, Parkinson's, and dementia. This compound has been found in the food supply chain in Guam and detected in the brains of individuals as well as American Alzheimer patients. And BMAE is suspected of causing neuron damage by promoting excessive glutamate X cytotoxicity, which has propelled its study as a significant environmental factor in ALS research across various preclinical models. Now, zebrafish are increasingly recognized as a preferred model for preclinical drug research, particularly in the study of neurodegenerative diseases. So we've partnered with Kremco Labs, a CRO with specializing in zebrafish studies, to conduct an assay involving neurotoxicity induced by BMAA. In this study, first we had to establish the toxicity parameters for BMAA using zebrafish larvae, and then we validated the study using Neuron, a therapeutic drug for ALS known to restore motor function, ensuring the relevance and the reliability of our findings in the context of ALS research. Our objective in this overall project is to develop a botanical drug. This is going to be a product composed of plant extracts that meet our targeted ingredient ratios. But as we finalize the chemistry manufacturing controls for this investigational product that we'll put into human clinical trials, we used botanically derived isolates at optimal ratios to ensure that in these initial tests, we were able to assess the overall target activity of our formulation just for these studies. With these materials, we then had to make sure that they actually worked in the medium. So what we determined was that an addition of 10 millimolar of cyclodextrin solution was safe and non-toxic as a carrier for dissolving the ALS formulation into the test medium. Our toxicity assays conducted 6 days post-treatment confirmed that all the products that we were using were safe up to a final concentration of 2 micromolar, and they all stayed in the medium. After we were able to do all this, it was time for our experiment. Our findings were able to show us that our CNR401 and some of our other products were able to demonstrate a statistically significant effect in this model, indicating the promising therapeutic effect that we have for potentially treating ALS. Treatment with our 401 in the zebrafish larvae led to a statistically significant improvement in the BMAA-induced phenotype compared to the positive control, Neuron. And these experiments were conducted in 48 well plates with 6 to 9 experiments performed per condition. After we knew we had our hit, then we wanted to test the efficacy of our formulation across various doses. So we did a couple more experiments. These assays were performed in 48 well plates with 7 replicates involving more than 60 larvae for each formulation. The results showed a dose-dependent response for both CNR401A and 402A, our mixed and pure formulations, respectively. So next, what we're doing is we're progressing our formulations and development research initiatives. Our current efforts are guided by the Botanical Drug Development Standards, which need us to refine our extraction SOPs, enhance our product fingerprinting, and ask us to adhere to rigorous QA/QC procedures. In terms of our drug delivery, we're focusing on optimizing our investigational product formats. Our pharmacokinetic studies are particularly concentrated on enhancing the blood-brain barrier penetration using things like solid lipid nanoparticles. And in addition, we're also embarking on a study of our investigational product in a canine degenerative myelopathy study at the University of Montreal. This is a naturally occurring animal model that mimics ALS and has various similarities because these dogs also carry the SOD1 mutant gene. We're also working to better understand the multi-target modes of actions of our formulation and what that has on ALS progression. One of the things that we're looking at is investigating the potential role of Cannflavins on modulating calcium levels in the brain and addressing impaired glutamate uptake by astrocytes. This is a hallmark of neurodegenerative diseases like ALS, so we're very interested in what we're going to find there. We're also doing some biomarker discovery, including RNA sequencing from our previous experiments to gain insight into gene regulation and biomarker analysis. And we're using bioinformatics screens from people on our team to enrich patient data profiles and predict outcomes by integrating existing data respiratories like Answer ALS, and other information so that we can identify biological pathways for intervention. One of the targets that we're really interested in is the bitter taste receptors, and we're going to talk more on this at our later presentations later in the year. We're also preparing and raising money to go into a pivotal phase 2 trial. Right now, we're putting our team and our infrastructure together, targeting a first patient dose sometime in the first 9 months of 2025. So we have a strong understanding of the roadmap in front of us, with a potential accelerated approval obtainable and orphan drug designation protecting our commercial advantages as we continue to grow and develop out this asset. Our patents are filed globally and give exclusivity to our methods, our compositions, and our products over the next 20 plus years. And beyond CNR 401, we also have multiple other assets in development that follow closely behind. And beyond this, we have numerous molecules in discovery, some of them that have been characterized, some of them that are yet to, that as we learn more and more about their therapeutic potential, we're extremely excited about what we can bring to the world. So, just as Oppenheimer and his team orchestrated reactions to change the course of history, today we're aiming to coordinate a therapy that can fundamentally change the trajectory of those living with ALS, its treatment, and its care. This isn't just an addition to our arsenal against ALS, it's a really transformational solution that will resonate across the spectrum of neurodegenerative diseases, bringing new hope where there were previously very little. So as we dive deeper into our work and we roll up our sleeves and get ready for the real work to begin, I invite any of you that may be able to help in any way possible to join our team, our story, which is a tale of groundbreaking science, complex challenges, and the profound impact of natural innovation. This is the moment we seek to emulate in our journey towards a breakthrough ALS treatment and a world without complex degenerative terminal illnesses. Thank you for listening to our presentation. A special shout out to our Caenorhabditis research team, all of our service providers, our partners, and the Government of Canada for providing funds that we were able to use to do this work. Thank you. And I have a couple of minutes, so if we have any questions, I'm here. Any questions? Or you can find me question? 2, Bonnie?
Thank you. Very interesting presentation. Oh, thank you so much. Very interesting. It's probably proprietary, but any chance to give us a hint as to what cannabinoids or non-cannabinoid compounds you're focused on?
As Ethan spoke about yesterday, we have a variety of different ingredients in our mixture that we know that do various things that help with the ALS pathophysiology. If you're at Ethan's presentation yesterday, you got a sneak peek, or you can bother Ethan later to find out about his trade secrets.
Hi, Akeem. Thank you. That was a great presentation. I'm curious about the results of the zebrafish model and I forget what drug it was that was the standard you were comparing against, which obviously did not translate to humans. And I'm curious about, your thoughts on the translational potential there and the results of your compound versus that drug.
Mm-hmm. Well, actually, Endeavorone is a solution right now that's used for ALS patients. It's approved as an IV formulation, but it only minimally extends lifespan, about 2 to 5 months, right? So we know it works at restoring motor functions, but what it doesn't do is, over a series of time, is stop the disease progression and help the patients. What we know is that with cannabinoids, our ingredients and our mixtures will be safe for chronic use, and there's a variety of different information on the various cannabinoids, terpenes, and flavonoids that we have in our mixture that should allow this to be beneficial for the patients at mitigating their symptoms, but also provide the neuroprotective potential, so that as they use it each and every day, they're able to stay alive longer, have a better quality of life, and it just changed their overall experience of what it means living with ALS or other motor neuron diseases.
Thank you. Great answer.
Hey there. Just wanted to say my grandfather passed away of ALS, and I just wanted to thank you for the critical work you're doing to helping sick people. Anybody in here, if you can help this guy, you should learn everything about what they're doing. We appreciate him.
Thank you. Thank you. Okay. Anyone else? If not, you can find me on lunch. I'm happy to chat. Thank you all.